Welcome to our YouTube channel of Applied Forensic Research Sciences. My name is Shalini Sharma. I am a volunteer in AFRS. Today's topic of presentation is different branches in forensic biology. So let's begin with our topic. In the upcoming presentation, we will be discussing about what is forensic science, what are the various branches of forensic science, what is forensic biology, what are the various branches of forensic biology including forensic serology, forensic odontology, forensic DNA fingerprinting, forensic entomology, forensic anthropology, forensic botany and forensic pathology. Now let us discuss about what is forensic science. The word forensic is originated from Latin word forensis, which means the forum, that is the court. Forensic science is a branch of science which involves various techniques and methods of sciences in the criminal justice system. The following mentioned are the major branches of forensic science, which are forensic chemistry, forensic toxicology, forensic biology and forensic serology, question documents and fingerprinting, forensic psychology, digital and cyber forensics, forensic physics and forensic ballistics. Moving forward in this presentation, we will focus majorly on the forensic biology. Forensic biology involves various methods of biology in the criminal and civil cases. In forensic biology, various biological fluids such as blood, saliva, urine, semen, sweat, etc. are analyzed. Blood typing was first introduced by Karl Landseer in 1901, who classified various blood groups into blood group A, blood group B, blood group AB and blood group O. Various tissue samples and various hair samples are also found on crime scene, which are analyzed for identification purposes. The following are the various branches of forensic biology, which are forensic serology, forensic taphonomy, forensic DNA profiling, forensic craniometry, forensic botany, forensic pathology, forensic microbiology, forensic odontology and forensic anthropology. We will be discussing more about them in the upcoming slides. Firstly, we will discuss about forensic serology. Forensic serology is the branch of biology which involves examination, detection, identification and individualization of biological fluids such as semen, blood, urine, fecal matter and saliva. Blood is most commonly recovered and one of the most important pieces of evidence utilized in the forensic investigation. It is commonly recovered in the cases of homicide, assault and terrorist attacks, including bomb blast cases. Blood as an evidence holds significance in the criminal justice system as it can link a crime with the criminal. Moreover, the pattern of blood stains can help in reconstruction of crime scene. At the crime scene, it is important to establish the type, origin and other characteristics of blood stain. Primary investigation in this regard are to confirm whether the recovered fluid is blood or whether it is of human origin or any other origin. Once the human origin is confirmed, it is analyzed to establish other characteristics. The blood stain is initially subjected to blood typing, where the proteins, enzymes and antigens present in blood of an individual are also analyzed. Forensic botany, an interdisciplinary science that combines an array of disciplines concerned with plants and fungal materials. Botanical evidences can provide the basis for linking a suspect to the crime scene. It links objects found on the crime scene to victim or suspect, which determines the place and time of incident and the place of stay before death, and also determining the origin of food or drugs, etc. It is used in solving criminal cases like kidnapping, murders, etc. It was first used in 1935 in Lindbergh case. It also helps in determining the cause of death of victim. Forensic odontology is a subspecialty of dentistry that has main focus on the identification of deceased person. It involves examination, handling and evaluation of dental evidences in a crime scene. It is used as a prominent method for identification in mass disaster situations. Forensic odontology brings knowledge of orofacial structures, their variations between people of different ancestry, and the implication of dental treatment to the identification process. Dental evidence is used in establishing human identity by comparing the dental features of a deceased person with the antemortem dental records. Dr. Asker Amido is considered as father of forensic odontology. Its first case was reported back in 1898 during the fire break in Paris, France. Forensic pathology is a subspecialty area of pathology that is specifically involved in the investigation of sudden, unexpected, or violent deaths. Thus, the practice of forensic pathology involves applying forensic scientific and pathological techniques to the investigation of lethal episodes. Forensic pathology can also be considered a part or a larger discipline of forensic medicine that applies forensic science to medical issues. It was started in 1877 by American Board of Pathology. It is also termed as an application of medical jurisprudence. And Dr. Rudolf Virchow is also known as the father of forensic pathology. DNA profiling is a forensic technique in the criminal investigations, which is used to compare criminal suspects' profile to the DNA evidences for assessing the likelihood of their involvement in the crime. It is also used in the paternity test to establish immigration eligibility and in genealogical medical research. DNA profile is also used in the study of animal and plant populations in the fields of zoological, botany, and agriculture. The first step in DNA profiling is to procure relevant DNA samples. Only a small amount of cells from a person's skin is needed to build a unique DNA profile. DNA is often discovered at the crime scenes during the police investigations. Once a sample is obtained, the forensic scientists extract DNA from the cells in the bodily tissues and then they separate the copied markers using the process known as capillary electrophoresis. Then they identify the distinct markers and they read the data by using the electropherogram, which plots the fragments of DNA and show how many repeats there are for each marker and where they occur. CODIS is the DNA database which is maintained by FBI for recording the DNA samples of criminal cases. Dr. Alec Jeffrey in 1984 had done notable work in DNA forensics. Forensic microbiology is involved in the examination, analysis of microorganisms or the toxins released for them. Sir Bernard Spilsberg is considered as the father of forensic microbiology. Forensic microbiology plays an important role in the identification whether the biological agent has been genetically manipulated or treated chemically to make it more virulent or to mask its original characteristics. In microbial forensics, law enforcement may partner with scientists from microbiology, genetics, public health, agriculture, and many other disciplines to identify and generalize the pathogens or their toxins implicated in biological events. Forensic entomology is the study of arthropods, especially insects, which are associated with crimes and other aspects of courts and judicial system. Forensic entomology usually involves the identification of insects and other arthropods associated with human remains as an aid to determine the time and place of death. Time of death can often be ascertained on the basis of ambient temperature and other weather conditions over the preceding days at the crime scene and by correlating this information with the developmental rates of arthropods on the corpse. These arthropods are typically the fly larvae, which are mostly the primary or secondary decomposers of animal remains. First systemic study on forensic entomology was conducted by Hermann Reinhard in 1881 It also helps in determination of primary crime scene and the presence and positioning of any wound present on the corpse. Craniometry is the branch of anthropology, and as the word suggests, it is related to cranium, which means the bone inside the head. Craniometry is used in the field of forensic science in order to gain information about the individual in cases where human skull is identified. The process of identification of human skull involves the identification of firstly the species, which is followed by the sex, age and race of individual. The study of human skull helps in the identification with the complete knowledge including the morphological and anatomical structures, bones, craniometric landmarks, sutures, specific features like the location of foramen magnum, orbits, types of teeth, and many more. Craniometry is widely used nowadays for identification in the field of forensic science in the identification of unknown individuals throughout the help of skulls. Samuel George Morton is regarded as the father of forensic craniometry. Forensic anthropology is the examination of human skeletal remains for law enforcement agencies to help with the recovery of human remains. It also determines the identity of unidentified human remains, interpret the trauma, and estimate the time since death. Forensic anthropology uses the technique of physical anthropology to analyze the skeletal, badly decomposed, or otherwise unidentified human remains to solve cases. Forensic anthropologists can assess the age, sex, and unique features of descendant and are invaluable in documenting trauma to the body and estimating how long a corpse has been decomposed. They focus on human skeletal traits such as skull features, dental characteristics, and subcranial bone sizes and shapes that vary from individual to individual and from population to population. In 1894, Thomas Dwight wrote articles and gave lectures on human identification in US and is considered as father of anthropology. Forensic taphonomy is the study of what happens to the dead body between death and recovery. Anthropological methods will work best when it is applied to bones that are well preserved, but the skeletons are easier to recover from the ground if they are complete and in good condition. Unfortunately, in all cases, it is not possible. There are a wide range of factors that affect the quality and quantity of bone that is recovered in forensic investigation. And here, the taphonomic factors play a huge role. Forensic taphonomy was introduced in 1940 by Ivan Efimov to study the changes in dead body while decomposition from biosphere to lithosphere. It comprises of 5 stages, which are disarticulation, dispersal, accumulation, fossilization, and mechanical alterations. Cause of death, manner of death, and time since death can also be determined by examining the dead body by the methods of forensic taphonomy. Now we will discuss about some of the forensic significances of forensic biology. Forensic biology helps in the determination of time since death of corpse. It is also used to link suspect to the crime scene. It also determines the primary crime scene. It is also used to determine the cause of death with manner of death. And the DNA profile of disease can also be determined. Thank you everyone for listening to this topic. I hope you have learned something productive from this. If you have any doubts, kindly comment down below. Please like, share our video. Also subscribe to our YouTube channel to learn about some more topics.